All right, let's get started then. All right, let me pull up stuff. Great. We're good to go, Dan?
Yep, good to go.
Sounds good. Well, good afternoon, everyone. Thank you so much for joining us today. Happy Friday. It's a beautiful sunny day in Seattle, and I'm delighted to be helping moderate today's Grand Round session. My name is Hao Tong, and I'm the inpatient chief resident at the University of Washington Medical Center Montlake campus. Today, my job is quite short. I have Dr. Abkowitz, the head of our division of hematology, helping introduce our Finch endowed speaker, Dr. John Dick. I'll hand it off to you, Dr. Abkowitz.
Thanks so much. So it's my pleasure today to first say something about Dr. Finch, and then to introduce Dr. Dick, who's our Clem Finch visiting professor this year. So Clem was the first division head for the Division of Hematology, and he served in that position for 32 years, which is quite remarkable, starting 74 years ago. He actually began at division head when he was 34 years old after completing his postdoctoral time. So it's clear that our administrative routing is a little bit different since his generation. And this is a picture of him when he first started as our division head. He is known incredibly well, internationally and nationally as well, for many contributions to very basic science and also clinical medicine of hematology. For example, he defined iron kinetics and the physiology of iron using iron kinetic and uptake studies, and studied the transferrin biology and actually predicted the existences of mediators like ferroportin and hepcidin and erythroferron just by understanding the biology of those molecules and what they should do. He said they should exist. And then many years later, of course, they were found and cloned and their functions shown to be exactly what he had said. He described red cell development and the physiology of anemias, including iron deficiency and ineffective erythroid production. He showed that hemochromatosis was due to the hyperabsorption of iron, which was quite a radical thought at the time, and it could be treated with phlebotomy. He defined many better ways to store blood, and he helped the World Health Organization understand nutritional anemia and was very active in the developing world and championed iron supplementation in food. In fact, between 1959 and 1980s, he consulted in studies of nutritional anemia that was conducted by the WHO around the world and traveled extensively. Some of the countries that he visited at that time and contributed to their nutritional programs are listed here. And he was awarded the 5th annual Bristol-Myers Award for Distinguished Achievement in Nutrition among his many scientific awards that have been given to him nationally and internationally. And this is actually a picture from Gambia in 1985 on one of his studies. So my memories of Clem, I actually knew Clem. He was the one who recruited me as a fellow to the University of Washington. He is very tall, both in size and certainly in stature, extraordinarily smart, very creative, an excellent observer and listener, and eclectic in all his interests, and particularly a creative problem solver that looked at anything, whether it be physiology or clinical medicine, and kind of figured out the why behind it. And so it's with that I'm very delighted to introduce our Clem Finch visiting professor this year. We've had a Clem Finch visiting professor since 1983 as a yearly occurrence. And we keep them quite busy because they do your medical grand rounds, our hematology grand rounds, a blood club, which is a community and an academic dinner on last night, and then meet with our fellows and meet with our trainees of all sorts and with faculty members during the 2 or 3 days with us. So John Dick is a senior scientist and holds a Canada Research Chair in Stem Cell Biology at the Princess Margaret Cancer Centre in Toronto, and is a member of the Toronto faculty. He's recognized internationally for his role in characterizing normal hematopoietic stem cells and also leukemia hematopoietic stem cells, and actually is a person that really pushed the concept that there is a cancer stem cell and what that means. He was the one who developed in his actually postdoctoral time what a xenograft is, so using human cells and putting them in mice and being able then to track the physiology of human hematopoietic stem cells or cancer cells in vivo in a murine situation and do experiments and figure out their properties. He's defined hematopoietic stem cells and isolated them both from a standpoint of flow cytometry and also multi-omics. And he's going to be talking about a bit of that today and some really novel ideas he has, particularly about CHIP and about some of the interactions of hematopoiesis and hematology with other fields within medicine. So I'm just delighted to introduce them to give today's topic on what makes a stem cell, and why does it go bad with age?
Great. Let's do the computer swap here. So lovely to be here, and thank you so much for the invitation and being able to give this illustrious lecture. So I don't have any disclosures that are relevant. So this is the talk that I wanted to present to you. So as Jan mentioned, we work on two sides of the same coin. So we work on the bright side, which are normal hematopoietic stem cells, and we also work on the dark side, which is leukemia, and where leukemia originates and what are the drivers of leukemogenesis. So, but today, for a more general audience, I thought that I would talk about our work on normal stem cells and how we've taken this approach and how it's led us in avenues and directions that are actually relevant, beyond hematology to cardiovascular disease and other things. And so we're trying to go through some of these different areas. So broadly, I want to talk about these sort of three things. What does a human stem cell look like? How do we assay them? How does the blood system change with aging? And then what is this link between inflammation? And a lot of what I'm going to talk about today is actually unpublished work. You're kind of the guinea pigs, actually one of my earlier presentations of this work already. So we'll see how we go. So just to set us all on the same page. So, as Jen mentioned, at the end of the day, the difficulty in studying human stem cells is that we can't understand, assay, grow hematopoietic stem cells in a lab dish. It's really difficult to study them. And the only way we can study a stem cell, assay it, is by watching what it does. And what it does is to regrow the blood system. And of course, is the armament of bone marrow transplantation, stem cell transplantation, and this, of course, is the center of that world. Don Thomas's work, Reiner Storb, and others contributed to that for the world. But of course, nobody wants to volunteer for my experiments, and so it's hard for me to find assay subjects to study human stem cells. And so many years ago, we figured out that we could take immune deficient mice. And because they don't have an immune system, they won't see the human cells as being foreign. And there's enough complementarity between what a human cell needs and a mouse can provide. And so we now have a mouse running around the cage if we transplant it with human hematopoietic stem cells. They can be cord blood or adult marrow or fetal liver, and they'll regenerate human blood. And so that led an odyssey, if you like, of almost 20 years. We did that work in the late 1980s, and we then 20 years later, we essentially worked out from single cell resolution what kinds of cells had that ability. And we found the 1 in a million cell, and it had this phenotype here, CD4-NF and a whole bunch of flow markers. And when we transplanted them into our mice, 1 in 3 or so of these cells had the ability to regenerate multi-lineage engraftment. Now, we looked at other cells there, and you can see now I have 3 populations of cells here. They're all stem cells in the sense that they all will give multi-lineage engraftment. They all will repopulate, at least in the xenograft assay, but only, these ones down here, the MPPs do it transiently. These ones do it a little longer. These ones do it longer, but they have the key property of being able to do it again. They have the ability to self-renew, which is the hallmark property of a stem cell that distinguishes it from all other cells. And so we had the approach then to try to understand these cells and determine what their mechanistic understanding was. One of the things we learned was, for example, that these cells are all dormant, but when they get activated into cycle, this cell takes 6 hours longer to go from G0 to G1 compared to this cell. And we figured out that it was controlled by a marker called CDK6. That actually becomes a very canonical marker for us to distinguish this difference between how a dormant cell gets primed and undergoes an activation cycle. So some of the things that we've learned over time was to understand this transition. So these are both long-term hematopoietic stem cells. So one's quiescent, the other one has started to think about dividing. It's being primed. It hasn't actually executed a division. And so we learned a lot about the 280 regions of the genome that are open in a dormant cell that have to get closed down in order for this transition to happen. If you prevent that from happening, you don't make this and the cells stay quiescent. So we know a lot about the epigenetic regulation. I told you about cell cycle. This is another paper we published a few years ago. This is not a homogeneous cell. Actually, there are cells here that when you transplant them in a mouse, some of them get going quickly and they start to generate the graft. Other ones take a while to get going. But interestingly, those that get going quickly, they do it at a cost. And the cost is that they lose stem cell function over time, where these ones will take a time to get going, but they maintain stemness for the long term. And this is actually very reminiscent of what happens in humans, which we can track in gene therapy trials. And then a new story on how organelles are, quiescence isn't just cells that don't divide in some kind of a silent cell. Actually, it's a programmed state. And one of the programs is that these cells have very active lysosomes. So they're constantly clipping receptors off their cell surface. And that's actually a way that these cells can't signal. So it's another way of maintaining dormancy. So I think I just went through this slide, so I'm just going to skip over this talk. So the point being here that, we used to think that this is a homogeneous population until we looked at it in a different context. And then we realized that there's heterogeneity, and we could actually prospectively isolate these cells. So there wasn't just a stochastic state where some cell did this and another cell did that. In fact, we could prospectively isolate these cells in CD112 low that had this property, CD112 high that had that. So unexpected heterogeneity. This is going to come back in a moment. So I'm going to talk to you. So why is this relevant? Well, here's just one clinical application. So our bone marrow transplant head, Smatson, came from the Karolinska a couple of years ago. We were talking to him, and Terrence is the head of the clinical flow core that monitors these patients. And what they were looking at was different clinical settings where we can begin to look at some of these new ways of assaying stem cells, because the approaches that we're using are not actually used in the clinic. They're a more rudimentary approach. And so obviously I don't need to tell you the importance of allogenic transplantation. It works in many cases, but sometimes, of course, the disease comes back. That's one situation. It also doesn't work sometimes when there's rejection and there's no graft, but there's this other setting where you have complete chimerism. So the graft is happening, but it's not functioning very well. And the clinicians call this poor graft function, PGF. And the problem is that, what do you do with this? How do you identify patients that are in this case? And how many of them are going to respond to, the TPO agonists, so L-thrombopac, or who's going to need a second boost of their transplant to try to get it to work again. And the problem is they have a hard time predicting this. And so we thought that, we could maybe help them out by using some of our more high dimensional flow. So they put together a set of 12 patients. This study happened literally a month ago. So you're getting fresh off the top of the head or off top of our lab data. So they assembled 12 patients at around day 60 or so, where they had taken a bone marrow. And these are patients that are doing fine. They didn't tell us who was in what category. And then they had 12 patients that clinically were in this PGF category. And so we ran our high dimensional flow. And this is just one example of that. So this is a patient who is doing fine. And this is just our flow category. I'm not, I know this is a broad audience, but just simply to say, there's a bunch of lymphoid progenitors. They're in this quadrant. Our potent hematopoietic stem cells are in this quadrant. These are those downstream progenitors, and these are the progenitors that give called the multi-lymphoid progenitor, but these are the ones that are going to repopulate the lymphoid side. So very nice graft in this person. And there was another sample we looked at, lots of deficiencies in these, particularly in the lymphoid progenitors, very deficient stem cells, reasonable number of MLPs, probably why this person was survival on this population. And so we did this on our patients. We categorized them just blindly. And so these are the patients we predicted as being controls. And these are the ones we predicted as being PGP based on these various markers. And sorry, just go back for 1 second. And so you can see we actually did pretty well, but not perfectly. And I'll come back to that in a minute. There's some here that we said were controls, but they were actually PGP patients. And there's some here that we classified as controls, but we classified as PGP, but they're actually controls. And this is an interesting one. So by looking at the actual stem cells, there's a patient here, which is PGP, but it seems to have very high stem cell numbers. So it turned out that when you look clinically, this is what happened to these patients. This is what I just finished telling you. So there's a subset. These are the controls. They're doing fine at day 60 or so. Here's the ones that were defined as PGP. One patient, C1, actually spontaneously recovered. The rest, 11 of them got L-thrombopac. Some of them responded really well, and some of them didn't. Some had a partial response. And so if you just look at, C1 here as an example, and I think I have them traced out. Oops. Okay. Sorry, I'm missing something here. So here's C1. So C1 is this guy that had the very high stem cell number. And so it turns out that, yes, it's PGP, but actually spontaneously recovered, and probably did because it had very high numbers of stem cells in them. And then this one here that we thought was a PGP cell or a control, but was actually a PGP, actually recovered really well on a thrombopack. So, we need more information clinically, and we obviously need a lot more patients, but I think this is promising to say that when we have a more refined view of stem cell numbers, we actually can diagnose and help patient monitoring. And this is going to be true for, the inherited anemias and a number of other settings where I think having a more high dimensional view of these flow markers will be actually quite valuable. Okay, so the next sort of part is, like everyone, we have a population, we think are stem cells. We need to figure out what makes them work, what makes them tick, and we want to carry out, mechanistic studies. And of course, the great way forward now is to take RNA sequencing, and we can use single cell RNA sequencing. So we have a gene expression program of every cell in the hematopoietic hierarchy. And the problem, and there's many papers on this, the problem is that many of the papers on human bone marrow were done on quite bulk cells. And the problem is stem cells are infrequent, one cell in 100,000 or so, depending on how you do it. And so even if you have 300,000 cells, there's actually very few numbers of cells that are actually hematopoietic stem cells and not very useful. And so we thought we wanted to do this in a more refined way. And so Andy Zhang is an MD PhD student in the lab, and he took the task of taking a bunch of public data, but it was enriched for CD34 positive cells where the frequency of stem cells is much higher. We gathered a lot of cells. And the trick here, of course, was to use advanced computational approaches where we could normalize the data properly and harmonize this information so that we're not getting overcoming batch effects and so forth. And he generated this new map, which is, got nice labels on it and so forth. The question is this real? And how do you validate? Well, the beauty in the blood system, of course, is that there's, vast amounts of data generated over the last, decades on highly sorted populations and their functional readout in whatever, erythroid lineage development, B cell, granulocytic, and so forth. And so we could take that known data and known gene expression signatures and map it on. And so this what you're seeing is a distillation of that. And here's just one example of that. And let's say, focusing on the stem and progenitor populations. So we want to know, well, how well do we do? And so this is a canonical stem cell sorting panel. And you see most of the cells land here. So this is a published study, single cell sequencing, a couple of, 1,000 cells, and they all land there. Here are the cells a little further down, and that's the trajectory they go. Here's what happens down the erythroid pathway, the megakaryocytic lineage. Here's the lymphoid pathway. Here's the early pro B cells, and here's the myeloid granulocytic macrophage pathway. So everything is fitting well, and this is just validation of how we generated our map. But of course, what we're really interested in are the stem cells, and I want to really see what we've dug into here. And so what we're showing here is the different kind of markers that one uses and assembles, and the field has assembled over the years, and we want to see how well do we do as we increasingly get to more and more purified populations. The frequency of stem cells in CD34 is perhaps 1 in 5,000 cells or so. And so you would expect that cells are going to be in that window, but they're going to be somewhere else. And so here are some examples. And then I'll just tell you, so just to orient you. So this is the marker. These are the sources, bone marrow versus cord blood. And then these are the papers that underlie the data I'm showing you. So we pressure tested our data as best as we could. And so here we are going with, lineage negative. This is now cord blood, a little more focused here. And now if you start to go in with more markers, so this is now the famous marker CD38, which really starts to drill down into cells that are really down in this little pod at the end here. This is our own data, many thousands of cells, and we add another marker, CD49F, 90, and then 49F is that canonical marker I showed you that we discovered that gave us the 1 cell in a million at complete purity. And this is unpublished data, but this is 5,000 highly sorted LTHSC of that phenotype, and they all land there. And so this gives us a lot of confidence that this map can be used as a reference. So anytime anyone has single cell expression data of bone marrow, we can look at all of it and say, okay, we're just interested in the stem cells. And so we just focus on the cells that land in this window. And so it's a nice computational purification. We don't have to go to a flow sorter. We can just purify it, if you like, based on this computational approach. Okay, so we're interested in these cells. So what can we do with that? Well, we want to know what makes them tick. And there's a lot of ways to do that, and independent ways. AI type approaches using NMF and independent approaches to see how good this pathway is. And so I had a student working on this, and we generated these maps. And so this is an independent way that basically says, here are a bunch of cells of the cell type. Here's a gene expression program. And so there's a bunch of cell types that align with, HSCs, a bunch of cell types that align with, all these different gradations of maturation you see here. These are the ones that will make Jan happy, orthochromatic, erythroblast, et cetera, et cetera. And so what you can do with that then is you can begin to, infer trajectories. You can say, well, okay, so cells start here and they end up going in this direction. So you can do a time analysis. You can figure out what are the programs that are changing? What are the transcription factors that are directing these different lineages? So by having a really high resolution reference map, you can really begin to extract what makes these cells tick in these different programs. So just to say we've done this is all very early data, but you can see along each of the lineages, you can get this trajectory. So we know not only where cells land in the map, but how did they get there and where are they going? And so we're obviously interested in the stem cell and then a homogeneous population, 1 in 3 cells repopulates on mouse. These cells should all be the same. But when we look at them, we realize that, what actually is lying underneath here. And so we took this, computational approach. I won't go into the details, but sort of a very approach where we took, many different sources of information. We took many different data sets and we just threw them in. We let, the computer tell us what's important. There's no direction in this data. And we ended up getting these, many signatures, and we distilled them. There are 17 meta signatures. So great, dots on a map. What does this matter? But what you'll see is okay. So I told you it's 17 different signatures, and we then investigated each of the signatures. The meta signatures down here, meta 1 to 17. Great, lots of dots again. But what we noticed was that what are driving each of these signatures? So you can see there's a bunch of genes here that are driving. Signature 11 is being very enriched for activated LTHC versus quiescent. Here's a quiescence. Here are some cells, signature 3, which is highly enriched for quiescent signatures. So these are all functional readouts and functional outputs, and they're highly enriched in each one of these signatures. And what you note, I will just point out, is here a whole bunch of signatures, from signature sort of 2 to about 7 or 8, and they're all NF-kappa B, inflammatory response. Why are purified stem cells having these inflammatory signatures embedded in them? So that's one. And the second thing I'll just simply say is, that dot of cells that by gene expression are landing in one spot, actually what's driving them are a whole bunch of different sub-programs. There's more heterogeneity in what we thought was a homogeneous population than we had anticipated. But just remember this inflammatory response. So that got us thinking, can we find, do stem cells respond to inflammatory responses? And this is a study done by Stephanie Shi, who's an affiliate scientist in my group, Andy Zhang. Murtaza Negri is the postdoc in the lab. And so I'm just going to give you, this is all unpublished work. And so just to set the stage, so these are, my quiescence activated stem cells. When these cells get going, the ultimate course is that they will differentiate, but occasionally these cells, of course, can self-renew and they'll go back into that state. And so we know that when you are exposed to an inflammatory setting, it turns out that not just your, B cells, T cells, and cells of the adaptive immune system are stimulated when they're hit in an inflammatory source. In fact, stem cells can do this as well. They respond to that. And there's actually beautiful work in the mouse system that Menno Passage and Eric Petrus and others have shown that in a mouse, if you get chronic stimulation, it's not just the downstream cells that respond, but stem cells are actually sensors of that. And one of the features of what happens with chronic inflammatory responses is the stem cells after chronic inflammation look like what happens in natural aging settings, or settings where these cells are, being abnormal and they're moving towards malignancy. So there's mirroring data on this, but there is nothing in the human system around this. And so we wanted to look at this. And so we asked, well, let's look at those inflammatory signatures. This is just another picture of this pure population. Here's AP1, this is inflammatory, here's stress targets. Here are the things we would expect, quiescence, activation, but this was the unusual stress signaling. And so we set up this study where we took cord blood cells, very naive population, threw them into mice, let engraftment happen. Then we hit them with a shot of PBS, TNF, or LPS. And then we looked 16 hours later. And what we got was exactly what you expect. This is now looking at engraftment levels, primitive cells, and so forth. And what you can see is these cells will decline markedly. So this is 16 hours later. This is the expected response. You're activating, stem cells are seeing that. And stem cells at 6 weeks are more cycling than stem cells at 20 weeks. We said, well, what about a more dormant stem cell population? Same thing. We waited 20 weeks. We hit them again, looked 16 hours later, and sure enough, we see exactly the same response. So there's the acute response. So what happens as a long-term consequence? What happens if we look at 2 weeks, one shot, but we look 2 weeks later, everything is recovered. So acute response, but then a little while later, everything recovers back to homeostasis. Exactly what we expect. But what happens, and there's no difference in the in the frequency. The frequency of stem cells is identical after these single challenges. But what happens if we do a dual challenge? So this is more like a more chronic kind of setting. We're getting one shot, we wait a couple of 8 weeks, we give another shot, and now we wait 20 weeks. So now we're looking 20 weeks later. So these cells have not seen their inflammatory response for 2.5 months, and look at the response. So a marked reduction in the functionality of these cells. What is interesting is if we take these stem cells at 20 weeks and we transplant them into secondaries, they still don't behave very well, but their number is essentially the same. In other words, there's the same frequency of stem cells, it's just they don't function as well after this dual challenge. And so these cells have some kind of a memory of a prior inflammatory response. I'll just skip this over. And so the question is, okay, so what is this? Can we find some mechanistic understanding? And of course, the only way we need to do this is by doing omic analysis. And we did actually multi-ohm, which is actually doing ATAC and single cell on the same cell type. And carry it on an analysis, and we get this is what the stem cells look like in a mouse. And so then we just did a computational approach where we just focused on cells that land in that window and asked, what did they look like? And the surprise was this. We ended up seeing two stem cell populations. We just call this HSC1, HSC2. We've never seen that before. This is the one that seemed to be, it's a link to all the other downstream progenitors. There's something about this one. And you can see that in mice that are treated with TNF 2.5 months before, you're actually driving this apart even further, and you see it even more with LPS. So this is the population which seems to be changing in the face of this dual inflammatory challenge. The question is, what is it? What makes these? Why are they different? And what is it that they're retaining about this? And so we carried out all the things that you would want to do. And the key thing we wanted to know is who are the drivers? Who are the transcription factors that are driving HSC1 versus HSC2? And we did it by either looking at ATAC, which you can do, or you can do it by, there are programs to do this by RNA sequencing. So completely independent, they came to exactly the same thing. And that is, if you look at HSC1 and you hit them with TNF, almost nothing happens to these cells. So it's like they don't respond, or they don't see it, or they recover, whatever. 2.5 months later, they don't look very much different if they've been seeing TNF. On the other hand, this HSC2 population, if you get TNF, you can see that it's changing, it's acquiring a lot of different transcription factors. And I'll show you in a second, all of the top factors here are FOS, June, AP1, things that are driving the canonical factors of inflammatory response. So it's like this second stem cell has a memory of something that happened 2.5 months before, and it all revolves around that inflammatory setting. And so there's the blow up of that setting, and so you can see all those factors up there. Oops. And so the question is, these cells aren't performing very well, and so we looked and we realized that there's one big difference between HSC1 and HSC2 is that these guys are actually been driven into quiescence. So these cells, after this repeated challenge, they, I think they seem to be driven into quiescence, which is why they're not functioning quite as well in the graft. They're still there, but they're not functioning quite as well. And so what this suggests then is there's two stem cell populations, and we just call this one HSC inflammatory memory population that retains memory of an inflammatory response. So this is all fine. It's a mouse system. It's a lot of computational. Doesn't have any relevance in reality. And so one of the canonical places of a memory signature, of course, is a T cell. Memory T cells are the canonical cell that responds to a prior memory. And so there's this beautiful study from Ralph Ahmed's group where they took a number of donors and vaccinated them with yellow fever. They fed them deuterium to monitor cycling cells. They captured the cells that had lost the label, were absolutely effector cells. And then they waited up to a decade or more, and of course found the heavy water positive cells. So they did respond, but they were absolutely memory. I think there's no other criteria that those are memory cells. And then they extracted them and obtained gene expression signatures of these cells. And so we asked, what does a long-term memory signature look like in relation to our hematopoietic stem cell memory? We made a signature of them here. And if you look at this signature and you compared them to these different populations, you can see that there's massive enrichment to T cell memory. So two very different cells are using similar kinds of mechanisms. And this is just the other GSEA of that same data. So that's one setting, okay? Maybe it's not just an artifact of our xenograph model. What about something real? Well, something we really have all exposed to, of course, is COVID. And we realized that there was data from Stephen Josefowitz from Cornell, who had acquired bone marrow data from patients who survived COVID. And the question was, what's the impact on HSC? So we contacted him and asked, we collaborated to get access to his data. And so he has this data where he has patients who had severe COVID, severe enough that they were in the ICU, and then they recovered. And they're 4 months after recovery, he obtained their bone marrow. So he took a bone marrow sample of these guys, had a set of patients who were in the ICU, also recovered, but not for inflammatory condition. They were there for other reasons. And then the healthy controls. And so we took their data. They have, of course, lots of bone marrow data. And we focused on the cells that landed in our stem cell window and asked, do we find any evidence of hematopoietic stem cell memory or not in those? And you can see, again, there's nice acquisition of a signature only in those patients that were exposed to COVID. So it looks like this is actually happening in the bone marrow stem cells of real patients exposed to a real infection, inflammatory control. So that's another setting. Well, the other great, setting is, of course, aging. And what happens to the signature with aging? We know that, with time, we're going to talk about clonal hematopoiesis, or CHIP, is something that happens. We also know that as we age, we're, of course, we count, or we have the sum total of the inflammations that we've had over our decades. And the question is, is the blood system changing? And do we acquire, if you like, HSC memory because we've had unique or more sequence of inflammatory events? And so we looked at some public data, and here is some pediatric bone marrow. So again, took this data, again, mapped it on our reference map and just focused on stem cells. And you can see that the inflammatory memory signature is much higher in young adults compared to pediatric patients. And then we have our own data. We obviously need more data here, but you can see 2 people at young adults, 2 people middle-aged, marked increase in the inflammatory memory, certainly compared to pediatric. And then it still is up. It goes down a little bit with elderly. We can talk about that, why that might be. But, generally, this is all much higher, obviously, than here. So inflammatory memory signature increases with aging. If you look at that, go back to that pathway, you remember these pathways here, that signatures 2 till 8, are seen here. We went back and looked at a variety of aging and our aging data, and you can see HSC memory is highly enriched for each of the signatures that involves, and it's only the HSC2, not the HSC1, is highly enriched in these signatures. So again, sort of independent data. And then here's the long-term memory signature. Again, independently, you can see is seen in the HSC2 and not in the HSC1. So everything is aligning that there is something unique about these cells. And then this is just looking, again, this is like an independent look at the data I showed you, which was gene expression signature. This is now this meta signature, and again it goes up with aging, and all these signatures that had this inflammatory memory are enriched through here. So it suggests then that there's something happening with aging, but then there's the other thing that happens, as I mentioned, clonal hematopoiesis. We acquire mutations with age, and the question is, what's the relationship between inflammatory memory and CH? And so here's an example of an experimental study where Irini Papiatrou has generated iPS cells using very similar approaches that Sabri here uses. Take human iPS and engineers in different mutations that represent the pre-leukemic MDS and then frank AML. And if you'd look, when she makes the changes, compares the normal to the first clonal hematopoiesis type clone in experimental setting, the most important factors that come up in hers are FOS, June, AP1, identical to what we see here. And so it looks like there's some relationship between pre-leukemia and this inflammatory memory. And so that got us to thinking about doing this directly. And so we collaborated with Paresh Vyas of Oxford, who has pioneered the development of TargetSeq, which is an ability to do single cell sequencing. So you get RNA data, but you can genotype the cells. And they do it by site-seq, so they can get immunophenotype. And so they have individuals who are known to have clonal hematopoiesis. So let's say between 2 to 15% of their blood system is coming from a stem cell that has picked up a mutation in TET2 or DNMT3A, and they compare them to age-matched patients who don't have clonal hematopoiesis. But then they can sort, and we can look at their stem cells, and they can say, this is a stem cell that has the mutation, and this is a stem cell that doesn't. And then we can ask, is there any difference in the HSC memory between these two? And the answer is that there's a huge enrichment. So the DNMT3A stem cells are highly enriched for HSC memory compared to stem cells from patients who don't have CH. So that's interesting, but what was even more interesting is this, and that is if you look at the wild type, that is the non-mutant cells in a patient who has clonal hematopoiesis, what about them? Turns out that they are also highly enriched. So the stem cell that has the mutation, but also the stem cell that doesn't bear it, is enriched for inflammatory memory. So how is that happening? Well, I'm going to wave my hands because I don't have any evidence for what I'm going to say next, but there's two possibilities, right? One is we know that TET2, for example, is a molecule which is quite inflammatory, and so it's possible that these inflammatory stem cells in their progeny are creating an inflammatory milieu, and they're impacting the other wild type cells that are present here. But the alternative explanation, which is perhaps even more interesting, is that if we age and our blood system is already changing and it's generating more inflammatory memory stem cells as the decades go on, and these cells are more and more dormant, more and more quiescent, they're less functional, you can see there's high selection pressure for these cells to acquire a mutation in 3A or TET that releases them from this restraint that that's been put on them, which is dormancy. And then, so it just means that all the stem cells that they have are inflammatory memory. One of them picks up a mutation, but the rest come out of the same pool. And so we really want to test that idea. This just tells you, this isn't just, we asked, well, how important is this inflammatory memory signature in this context? And so we assembled, the 7,500 biological pathways that are known in biology and asked, how enriched are they in one context? The number one hit out of 7,000 is this inflammatory memory. TNF signaling is here, which is very, very high, but this tells you how powerful this pathway is at defining this difference in state here. And so what we have then are two different populations, and I think this becomes really important for, thinking about things beyond, leukemia, clonal hematopoiesis, and so forth. How relevant is this for other kinds of diseases? Because we know that as humans age, many of the initiating events of the diseases of aging, from neurodegeneration to fibrosis and other kinds of features, have early on often an inflammatory component. And one wonders whether this is a component that might underlie some of this. And so, I think that we're very interested in assessing this in a variety of settings, as I'm sort of saying at the bottom of this slide. And so I'm just going to end off with just, one storyline that we've been working on. So, we had worked on clonal hematopoiesis and, the fact that this is setting up an inflammatory setting. And there is obviously the work of Sid Jaiswal, published in 2017, which broke this field open because, so of course we were interested and had the defining paper showing that a subset of people with clonal hematopoiesis will go on and get leukemia, but that's 4 per 100,000. But of course, 1 in 3 people have clonal hematopoiesis. And what Sid realized was that these people have a high propensity for cardiovascular risk. And so you can see that here. This is just the prevalence as time goes on, and much higher in patients with heart disease and arthritis and so forth. These are the two papers that we published with our clinical group, and I'll just go through these two. I'll just go through short studies, but so we have developed a clinical assay that's actually in our clinical lab, which is a molecular inversion probe assay to create a panel which is very inexpensive and very rapid for doing clonal hematopoiesis testing. And so both of these studies used about 1,000 patients, and we could do this very, easily and readily and at a quite, reasonable cost. And like I said, this is now deployed in our clinical lab at the Princess Margaret Cancer Center. That assay, I should say, is developed by Sagi Abelson, who is the driver of the development of the CH analysis in my group, and the clinical lead on this was Phyllis Bilia. So this is just the data from the JSWAL paper, just to remind everybody that if you have CH, you have a much higher risk of coronary artery disease. You have stroke risk. This is the various risk alleles, and you can see CHIP or clonal hematopoiesis, has a risk that's higher than smoking, for example. So it's a significant risk in CH. The one setting that we wanted to study, so Phyllis came to me when we talked to her, and I said, we should test something. And she says, we're not going to redo what was published in the JSWAL paper, but there's this other setting is with patients with heart failure. There's one set of patients who are at home and being treated medically. There's other people with heart failure who are in the ICU, and they're in this cascade of cardiogenic shock, which is driven by this inflammatory response syndrome. And, as they say, it's this death spiral, very high, I think 50% incidence of death in these populations. But the problem is they can't figure out clinically who's going to survive and who's not. And so how are you going to intervene? And so we, this is driven by Florence. And so we carried out this study. And so here are the data sets. So it was about 700 odd patients, clinically matched by all criteria. And if you just look at the 30-day survival, if you have clonal hematopoiesis, you do much more poorly than that's all told than if you don't. But if you look at individuals, you can see TET is really driving poor outcome compared to some of these other ones. And so there's something really profound happening with TET, and that's very significant. See the hazard ratio here. And this is the 3 years still holds. You actually drive apart these curves even more. And again, you really see the poor survival outcomes in a few of these drivers here. And so this is, the clinicians who are working with us are very interested in continue to use this and deploying it clinically to begin to make decisions. Now, the one thing that Phyllis had was that they have a large biobank in their cardiac center. They also kept plasma. And so they had looked at, and they had done cytokine arrays on these. And so we looked at different, cytokines here. You can see that, interferon levels, TNF levels, and so forth are much higher in patients that are TET positive compared to the control. So part of this must be driven by the cytokine milieu, which is being delivered by the TET allele or by the TET clone that's elaborating. But, there could be other players that are involved here. Now, these are patients that are in trouble, and a subset out of the 700, 100 of them went on to getting a heart transplant. PMH has a large heart transplant program. And so we looked at the patients. So, well, not 100, I guess there were 127 actually. And 100 of them didn't have CH, 26 of them did. And you can see here that there's actually, no different. They're all matched for these markers, but you can see here mortality is significantly different if you have CH than not. And so CH is already, predicting the success of organ transplant. And here's the data on follow-up survival in these patients. And so Phyllis is working very hard to work with the solid organ transplant group to begin to ask, they don't take into account the CH status of the individuals who are getting their lungs and livers and kidneys. And so we're setting up a big program to try to monitor CH in these solid organ transplants to see whether this gives a better strategy for trying to define people who might need different kind of monitoring because of their possibility of these kinds of directions. So just to close then on that, sort of, I move through that quite quickly, but I'm talking to you about hearts, but I think this is going to be true for many of the diseases of aging. And I think this is a new axis that needs to be investigated. But of course, the stuff I told you before on inflammatory memory is another axis to be investigated, which doesn't have this genetic driver, but has, we don't know what's driving it, but we think that's another axis. So I moved through a lot of different territory, so I'm happy to take questions, and thank you very much for your attention.
Wow, what amazing talk. I'm so humbled by your team's research and so appreciative of you taking your time out of your schedule, Dr. Dick, with us. And so we'll transition to our Q&A session now. Please submit any questions you have or discussion points you have in the chat below. I did see a hand raised from Dr. Dishek, and let me see. I am allowing Dr. Dishek here to speak.
Yeah, so since that was a wonderful talk, and since this is a general medical audience, perhaps I'll ask a general medical question. So there's this debate about whether inflammatory exposures early in life are good or bad, and whether being too clean with children is perhaps a danger. How does this impact on that? Because it would seem that having inflammatory exposures while young might predispose you to this clonal hematopoiesis and all its consequences when older. Have you thought about that?
We've thought about it. We have no data, so you're just going to get my hand waving. I think it's, so this is, Reese, we only uncovered this in the fall. So this is, 6-7 month old data. And so, there's a lot of things to do. And obviously one of the main things is we need to publish it so people can actually start working on it. But, we're talking about it, and we think that this is an X factor, I think, that needs to be investigated in many different settings. And what you're pointing out, I think, is one of those. And I think that it would be very interesting. I mean, the studies that are being done on genetic tracing of malignancies that arise when you're 30, 40, 50, there are some beautiful studies that have been published that said the initiating events are happening when the person is either a child, that clone was born then, or there are some few cases where it was actually born already in utero. And that it raises many questions in terms of our thinking on the disease progression. I mean, this is in malignancy, myeloproliferative neoplasm and so forth has been traced back that way. One of the things I can tell you, I could give you another whole talk on this, but we recently modeled Down syndrome AMKL. So this is the set of children who have Down syndrome, 25% or so will generate this transient leukemia when they're born, and then a subset of those will go on and get a leukemia sometime later in their childhood. And it revolves around GATA1S and STAG2. So the molecules are known, but the question is, how does this happen? So we actually took human fetal livers, we engineered the GATA1S into disomic fetal livers or trisomic, so from Down's fetal livers, and showed that we could only get this pre-leukemia, transient leukemia from that particular, not just a fetal liver cell, but a Down syndrome fetal liver cell. If we engineered the second mutation to get leukemia, we could get full-blown leukemia from either disomic or trisomic. But the interesting thing, and the reason I'm sort of saying it this way, is that I won 2 really good bottles of wine because I bet my postdocs, because they said, oh, let's try this. Because I told them, I bet this is developmentally specific. I bet there's something unique about the fetal situation. And this is why we're getting malignancy then, and we don't get it in adults, right? There's many kinds of, mutations that only occur in children. You never see them in adults. And so we engineered in adult marrow, and sure enough, it didn't work. We could not, even with 2 molecules, we couldn't get any leukemia established in adults. So there's something about the fetal milieu. So a long way to say, I think that context matters, and I think that we really need to understand these exposures and how the immune system is changing. And are there children who have a much higher inflammatory memory, which are at more risk for certain things? So lots of hand waving there.
Hey, Dr. Dick. How had a power incident, and she's logging on from a different computer. So if you want, are you able to see the chat? There's several.
Yes. Okay, here we go. Yes, I can see the chat. Okay. Let's see. Right. So someone was asking, do you think that HSC2 status explains why normal HSC are suppressed in MDS? So this is an interesting one, isn't it? So you have these impairments of normal hematopoiesis, when you have these, pre-malignant clones that are coming, and sometimes, the level of the pre-malignancy, MPN and so forth can be, not high, right? And yet they're creating an impact on the normal hematopoietic environment. And so, yes, one wonders, I mean, MPN is an interesting one. You're talking about MDS, but MPN, right? So we know JAK is very closely linked to, higher inflammation counts. And so, you can have a situation where maybe your VAF of the JAK mutation at some point in your disease is, 5%, 10%, but yet you can have, more profound impairment. Yeah, I wonder whether this is actually occurring. So, it will be difficult to assess patients on large scale the way we're doing it now, because the only way we can find HSC2 is by doing this, single cell omics. We need to look at the gene expression programs and stem cells. And so we're working hard to see if we can find, cell surface markers that we could use for a flow-based assay, which would make much doable of the kind of things that you're talking about here. But in principle, yes, I think that this is something which is really, really valuable, important. Do the progeny of HSC2 have altered function? I guess the only assay we have for that is when we did the serial transplant. So we had the 20-weekers, and we knew that these were less exposed to TNF, so they would be quite enriched for the HSC2. And then we did a serial transplant, and we showed that they're transplantable, but the quality of the graft in that secondary is still kind of impaired. So it seems to me that they do have some kind of altered function. So, I was talking to one of the fellows today, it raises this question. If you remove the inflammatory stresses that cause this, do the cells rejuvenate or repair? And at least at this context, it doesn't seem like it. Maybe if we waited longer time, or there's something about the milieu, but I think we're not sure about that possibility. Sorry, so there's a question here that you're speaking to a PhD, so wondering about CH and the risk of CKD, ILD, and IPF. What is that?
Chronic kidney disease.
Kidney disease. I don't know. Since I don't even know the acronym, I guess that's the telling part to the answer to that question. Any other questions? People want to chime in? I don't know if the hosts are allowing that, or you're only going through on chat.
We normally people can put questions in the chat, but if somebody wants to raise their hand, I can allow them to talk. I will let you know that there is a power incident at the hospital right now. So good thing you're over not at the Mall Lake Center. So the whole hospital's out of power, is what I'm being told. So
oh, wow, that's I'm sure there'll be many people scurrying around that incident. Okay.
If there is no more questions,
Thanks,
I can
everyone.
definitely go ahead and end the talk.
Great.
Thank you for joining us for Department of Medicine Grand Rounds. And yeah, we do appreciate your time. I normally don't do this, so you'll have to excuse me for the end. But if there's any other questions, I can make sure I send those to you as well.
Okay.
So,
super. All right. Thanks,
all
everyone.
right.
Goodbye.